So this is the story. Each chapter starts with a story, and I couldn't find a video for this story, so I just put it here. Talks about the King of Prussia wanted to build up his military. He was really obsessed with tall men to be in his military. He was really short. He was only 4'11". So he wanted really tall guys to make up his military. It was very expensive to recruit. So eventually he just went around to the towns and picked out the tallest women and made the tall men breed with the tall women. But that eventually became expensive, and it took a long time because it takes a long time for people to grow into adulthood and to their full height. And they never really turned out how he wanted them to. Just because two tall people get together and have a baby doesn't mean that baby is going to be tall. So in the second paragraph, it says, King Frederick's attempt to selectively breed tall men was a failure, would be universally condemned today for ethical reasons, like our eugenics movement that we talked about. You can't just breed people on purpose because you want certain traits. However, using today's genetic technology, it is possible to select embryos for specific traits and implant only those embryos into a uterus for growth in development. This is part of the in vitro fertilization process. If genetic markers for above average height, IQ, or muscle patterns associated with athletic success are discovered, tests can be used to identify embryos carrying these traits, giving rise to designer babies. If such tests were available, would you consider having such tests done and implanting embryos carrying such markers? Would you want your child to have a high IQ and good athletic ability? It's actually going to be part of your discussion in the between this week and next week. We're going to be talking a lot about genetic testing and designer babies. So that's introducing our chapter of complex traits. So what exactly went wrong with his experiments? After all, when Mendel intercrossed true breeding tall plants, the offspring were all tall. So why isn't it the same for humans? And even when the heterozygous tall pea plants are crossed, 75% of those offspring were tall. The situation in humans is more complex than in pea plants. We have more genes that control our height than the pea plants do. So we will be talking about King Frederick and his tall men throughout the chapter. So we're going to review some words. Genotype is a set of genes in your cells that you inherit from your parents. You get one of each. We use the letters to designate. They can be homozygous dominant, where they're both the same in capital. They can be heterozygous, where you have one capital and one lowercase. Or they could both be lowercase, which means they're both recessive. Phenotype is the physical characteristics that manifest due to the genotype. And then variation is the differences in physical characteristics among a population of the same species. Us humans, we are all the same species, but we have a variation in our eye color, our hair color, our height, our intelligence levels, our skin tones, everything. So these three words are going to be used a lot in this chapter. So to understand this chapter, you have to remember what these three main words mean. If you don't know what they mean, this chapter is going to be very confusing. All right. Phenotypes can be either continuous or discontinuous. Discontinuous are phenotypes that fall into the two categories. They're either or. They're controlled by a single gene pair. Being able to roll your tongue. How many of you can roll your tongue? That's actually a gene that you inherit from your parents, and you can either do it or you can't do it. That's a discontinuous trait. It only has two options. A widow's peak, some people have, where they, like Superman, I always think of when he has a little widow's peak where it goes to a point on your forehead, your hairline. That's a single gene trait. And then the way you clasp your hands, whether you go right over left, which way is more comfortable? You can go either way, but which way is more comfortable? And that's a single gene pair. These are discontinuous traits where they have either one thing or the other. Continuous variation are phenotypes that are distributed from one extreme to another in an overlapping or continuous fashion. They're controlled by several gene pairs or an environmental interaction. So sometimes it's just genes, Sometimes the environment comes into play, and height is one of these things. It's controlled by continuous variation. This is what King Frederick didn't understand about genetics in the 1700s. Is that just, like I said, just because you breed two tall people doesn't mean you're going to get all tall offspring. Height is a continuous trait where you can get a range of different heights. Monogenic versus complex traits. The prefix mono always means one. So monogenic traits are controlled by a single gene. Those are the first traits we talked about, the discontinuous ones. And then you have complex traits. Most of our traits are pretty complex. They're controlled by more than one factor. They're also known as polygenic traits. The prefix poly means many. So they're controlled by many traits. They're controlled by more than one gene. They can also be multifactorial, where they're controlled by more than one gene, and the environment influences them also. And human height is one of these genes. Many of our traits are multifactorial, where they're controlled by several genes and the environment. The distribution of phenotypic variation in a population for a polygenic trait follows a bell-shaped or normal curve. These are heights distributed in a population. These are just the averages. On average, you're going to have middle of the road heights. You're going to have a few really tall people and a few really short people. For the most part, everybody's going to fall in a certain range. And then there's an example using actual people, a bell-shaped curve. You have the really short people on one side, the really tall people on the other side. But for the most part, they're pretty average. In traits controlled by two or more genes, a small number of the offspring have phenotypes identical to the parental generation, either very short or very tall. But most offspring of that next generation have phenotypes that fall between the extremes. That's exactly what we just saw on that curve. Their distribution follows the bell-shaped curve, with each gene adding a small but equal amount to the phenotype. And two or more genes contribute to the phenotype. However, the contribution from each individual gene may be small. So just because you inherit these genes doesn't mean you will exhibit the phenotype to a certain degree. Phenotypes vary across a wide range, and their variation is best studied in populations rather than individuals. When scientists study traits, population or ecology scientists, they're usually studying an entire population of people or species. They don't just study one person or one family. They study an entire city or region or country. Traits such as height, weight, skin color, eye color, and intelligence are complex traits under polygenic control. All of these traits have many different genes that contribute to their phenotype. In addition, many congenital malformations such as neural tube defects, cleft palate, clubfoot, as well as disorders such as diabetes and hypertension, along with some behavioral disorders, are polygenic and or multifactorial. Sometimes the environment comes into play. Example, human skin color is controlled by 3 or 4 genes plus environmental factors, leading to a wide range of phenotypes. The greater the number of genes controlling a trait, the greater the number of phenotypic classes, or the greater number of traits you can physically see. Example, eye color is a trait controlled by 2 genes. Those 2 genes each have dominant and recessive alleles themselves. Individuals in the F2 generation, so remember you start with the parents have babies. That first set of offspring is called F1, that next set is called the F2 generation. Will display 5 phenotypic classes due to the number of dominant alleles they inherit. An example is eye color. If you don't have any dominant alleles, all your alleles are recessive, then you have blue eyes. If you have all dominant alleles, all 5, then your eyes are dark brown. That is how eye color is controlled. So if it's controlled by 2 genes, there's a certain ratio that you will see in that F2 generation, 1 to 4 to 6 to 4 to 1. And the classes are the eye colors. So there'll be 1 dark brown on one side, there'll be 1 blue on the other side, and then you have the mix in between. If you have 3 genes, you have an even larger ratio, but it's still a bell-shaped curve, where the average is somewhere in between the 2 extremes. 4 genes, same thing. You have the bell-shaped curve. There's a higher number of individuals in the middle, with the phenotype in between the 2 parents. So if you assume each gene controlling a complex trait has an equal effect on the phenotype, the effects of the gene are added together. So your example is King Frederick William's guards, Potsdam guards. So if you take a 6'9" man and breed them with a 6'3" woman, their gametes are below. You can see there are a chance that the child will inherit a lot of recessive alleles. The more recessive alleles they inherit, the shorter they are. So there's a chance that these 2 tall people will have short children. So they did the whole Punnett square, the dihybrid cross for it. And children of very tall or very short parents are usually closer to the average height of the population rather than the average height of their parents. This important concept is called regression to the mean. And this is why King Frederick William's attempt to breed giants for his elite guard unit failed. He did not know about the regression to the mean. or about genes or alleles or any of it. So using very tall parents results in more children with a height that is average between the parents than it does actual tall children. And then when you add in the fact that many of these guards are probably tall because of environmental factors, they had some kind of disorder or malfunction, and those disorders and malfunctions aren't part of their genes, they don't get passed on to their children, it's easy to see why his program was a failure. He didn't actually breed tall children. This is just a case study. Percentage of offspring. The percentage of offspring was somewhere in the middle. You had a few really tall, a few shorter. Most of the time they fall in the middle. Multifactorial traits controlled by 2 or more genes. Each contributes a small amount to the phenotype, but there are also environmental factors that control these traits. Environmental factors interact with the genotype to produce the phenotype. Epigenetics is a new field helping us understand and explain how our cells can selectively turn on or off different gene sets in response to environmental factors. And we're going to watch this video really quick. It's only like 4 minutes long. These changes in gene expression are mediated by reversible chemical modifications to genes. This makes more sense if you can actually see it.
Imagine coming into the world with a person so like yourself that for a time you don't understand mirrors.
As a child, when I looked in the mirror, I'd say, "That's my sister." And my mother would say, "No, that's your reflection."
It's hardly surprising because you both come from the same egg. You have precisely the same genes. And you're literally clones, better known as identical twins. And yet, it's not uncommon for a twin like Anna Marie to get diagnosed with cancer, while the other, like Clotilde, doesn't. But how can 2 people so alike be so unalike? Well, these mice may hold a clue. Their DNA is as identical as Anna Marie and Clotilde's, despite the differences in their color and size. The human who studies them is Duke University's Randy Girdle. So Randy, I see here you have skinny mice and fat mice. What have you done in this lab?
Well, these animals are actually genetically identical.
The fat ones and the skinny ones.
That's correct.
This gets even more mysterious when you realize that these identical mice both have a particular gene called agouti. But in the yellow mouse, it stays on all the time, causing obesity. So what accounts for the thin mouse? A tiny chemical tag of carbon and hydrogen called a methyl group has affixed to the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl groups, attach to genes directly, inhibiting their function. Other types grab the proteins, called histones, around which genes coil, and tighten or loosen them to control gene expression. Distinct methylation and histone patterns exist in every cell, constituting a sort of second genome, the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, how to work, and how much.
In fact, It's the epigenome that tells our cells what sort of cells they should be. Skin, hair, heart. You see, all these cells have the same genes, but their epigenomes silence the unneeded ones to make cells different from one another. Epigenetic instructions pass on as cells divide, but they're not necessarily permanent. Researchers think they can change. And that brings us to the reason why we're showing you twins. In 2005, they participated in a groundbreaking study in Madrid. Its aim? To show just how identical epigenetically they are or aren't.
Our genes are just part of the story. Something has to regulate these genes. And part of the explanation is epigenetics.
Estella wanted to see if the twins' epigenomes might account for their differences. To find out, he and his team collected cells from 40 pairs of identical twins, aged 3 to 74. Then began the laborious process of dissolving the cells until all that was left were wispy strands of DNA, the master molecule that contains our genes. Next, researchers amplified fragments of the DNA until the genes themselves became detectable. Those that had been turned off epigenetically appear as dark pink bands on the gel. Now, the genes from a pair of twins are cut out and overlapped. The results are far from subtle, especially when you compare the epigenomes of two sets of twins that differ in age. Here on the left is the overlapped DNA of 6-year-old Javier and Carlos. The yellow indicates where their gene expression is identical. On the right is the DNA of 66-year-old Anna Marie and Clotilde. In contrast to the younger twins, hardly any yellow shines through. Their epigenomes have changed dramatically. The study suggests that as twins age, epigenetic differences accumulate, especially when their lifestyles differ.
One of the main findings of research is that epigenomes can change in function of what we eat, of what we smoke, of what we drink. And this is one of the key differences between epigenetics and genetics.
All right, so that was epigenetics. We're going to talk about twin studies here some more in a little bit. So some complex traits do not show a typical continuous distribution. Individuals are either affected or not. Congenital birth defects, such as club foot or cleft palate, are examples of traits that are distributed discontinuously in a way that resembles an inheritance of a single gene traits, but are complex traits controlled by several genes. So if you just look at them from the outside, they look like they are single genes. They're either or, but they're actually complex if you look at it closer. And then there's a model that would explain this. They call it the threshold model. And risk for a disorder should decrease as the degree of relatedness decreases. So parent and child have a high degree of relatedness. You're very closely related. But second cousins, third cousins, when you spread out, the degree of relatedness goes down. So in the threshold model, the chances of getting a specific genetic disorder are distributed among the bell-shaped curve, just like the other ones. Specific genotypes have the potential, but only a limited number of genotypes actually express the abnormal phenotype. And the trait or disorder will only be present in those individuals with the limited genotypes if they are also exposed to the proper environmental conditions. So you have to have the genotype and you have to be exposed to the environmental conditions. And that's called the threshold. Once you meet that threshold, then you're affected. You have whatever disorder they're talking about or studying. Genetic counselors assess recurrence risk with multifactorial and complex traits. So there are people that go into this field to be able to counsel. Before they have babies, they go to these genetic counselors and they let them see their genomes and see what kind of disorders run in their family, and they talk about the risk of the children having any of these disorders. So the recurrence risk for an abnormal phenotype, somebody with a disorder, depends on something called consanguinity. Related parents have a higher risk of having a child with the complex disorder. That's a big, long word, but that's what it means. Another characteristic is if there's a previously affected child. Parents with 2 affected children have a greater risk of having another affected child, since their genotypes are likely close to the threshold. Severity of the defects is also something to consider with these traits. A severe phenotype indicates the genotype is well over the threshold and increases the risk of having another affected child. And sometimes there's higher frequency in one sex over the other. For complex traits expressed more frequently in one sex, the less frequently affected sex has a lower recurrence risk. Doesn't mean it's gone away completely. There's still a chance. But if it's higher in one gender than the other, then it's less likely that the other gender won't have it. To measure the interaction between genotype and the environment, we begin by examining the amount of phenotypic variation in a population rather than among individuals. Measuring it this way highlights the interaction between genetic and environmental influences on phenotype. I told you we were going to use these words a lot. Genotype, phenotype, variation. Phenotypic variation caused by the presence of different genotypes in a population is known as genetic variance. The proportion of phenotypic variation that is caused by different genotypes is called the heritability of a trait. And variation among individuals with the same genotype is known as environmental variance. So if there's different phenotypes, but people have the same genes, then it's likely due to the environment. The environment is causing those different phenotypes instead of the genes. Twin studies. The closer the genetic relationship between individuals, the greater the chance that their environment will also be similar. So to calculate the relative influence of genetic versus environmental factors, scientists study twins. They also study adopted children and siblings. These studies are used to separate and evaluate the genetic and environmental factors responsible for phenotypic variation. Identical twins that are separated at birth. They have an identical genotype, however, they have different environments they were raised in. So comparing those gives scientists a lot of answers. And then the opposite situation, adopted children living in the same family. They have very different genotypes, but they're all being raised in the same environment. So those are ideal conditions for scientists to study. Two different types of twins. There's monozygotic twins, abbreviated MZ. These are genetically identical twins derived from a single sperm fertilization event involving one egg and one sperm. So one sperm fertilizes one egg, and they happen to split into two. These will be your identical twins. These twins also, when they're in the womb, they usually share a placenta between the two of them. And then dizygotic twins, these are your fraternal twins, derived from two separate and nearly simultaneous fertilizations involving two eggs and two sperm. So these twins have the same amount of genetic information as just regular siblings. They just happen to be fertilized at the same time. So the mother produced two eggs that month, the two different sperm fertilized them. They each combined in their unique way. And they're siblings that happen to be born on the same day. These are fraternal twins. And there are quite a few sets of fraternal twins where one twin is very dark and one twin is very light. And the babies are a case of one being an albino baby. And their genes just got mixed up differently from their parents. They share 50% of their genes. For a complex trait such as height, if heritability is high, the variation in phenotype is largely genetic, and monozygotic twins will be closer in height than dizygotic twins. So the monozygotic twins share 100% of their DNA, their height, same thing, they will be the same. Height does have an environmental factor, but usually identical twins are raised in the same house, so they have the same environment. And then dizygotic fraternal twins are just like any other set of siblings. They inherit their own gene combination from their parents, and they won't always have the same height. And if heritability is low and variation in height is due to mostly environmental factors, then they all should vary in height together. More environmental factors are involved, then they all won't be the exact same. If genes are involved, monozygotic twins are the exact same. If it's more environment, then they won't be the exact same. Twins in concordance. To evaluate phenotypic differences between twins, traits are scored as either present or absent, rather than measured quantitatively. A trait is called concordant if both twins have the trait, and discordant, if only one twin has the trait. Identical twins tend to be concordant for everything. And fraternal twins are concordant for about 50% of their genes. And discordant for the other 50. The greater the difference in concordant rates between the two sets of twins for a given trait, the greater the heritability. For a trait controlled completely by genetics, the concordance rate should be 1 or 100% for the monozygotic or identical twins, and then 0.5 or 50% for the dizygotic twins. Concordance gets converted to heritability by statistical methods. And there's a table. Concordance values in monozygotic and dizygotic twins between blood types, eye color, mental state, hair color, Down syndrome, left or right-handedness. So you can see monozygotic twins, the concordance values where both twins have the trait are very high. Twin studies of obesity show a strong heritability component. Obesity runs in families. Genetic clues to obesity. There's a gene named it the OB gene, the obesity gene. So increases in the incidence of obesity have taken place in the last 30 to 40 years. It's unlikely that large-scale changes in our genetic makeup are responsible for this increase. We didn't all of a sudden start producing the obesity gene more frequently in the past 30 to 40 years. It's always been present. So changing environmental factors, including diet and physical activity, is interacting with our genotype somehow. So twin adoption and family studies show that there is a strong genetic component in obesity risk factors with high levels of heritability that average close to 70%. That is a very high rate of heritability for something like obesity. OB gene encodes the weight controlling hormone leptin in mice. You saw a little bit of this in the video. Binding of leptin to its receptor, which has its gene. The receptor has its own gene. And the hypothalamus controls energy consumption and conversion to fat. And there are a lot of environmental factors that control this. The faster you can convert fat, the less likely you will be obese. And these are both controlled by both genes and environmental factors. In human genes, like I said, there's two different genes, one for leptin and one for its receptor. Mutations in these and other genes account for only 5% of obesity cases, indicating that obesity is a multifactorial trait. So there could be mutations in these genes that are causing a few people to be overweight, but these are only 5% of the cases. And here's a figure. You can see all the chromosomes lined up. This is somebody's random chromosomes. There's some obesity genes in the human genome. The green triangles represent single genes in which obesity is the main phenotype with little environmental influence. Then there's red triangles. Genes are candidates for obesity genes. They're not sure yet, but scientists think they control obesity, or that they contribute to obesity. Mutations in these genes have an association with obesity. Red bars represent chromosome regions that may contain other genes, and more than 70 genes associated with obesity have been identified. So we have over 70 genes in our chromosomes and our DNA that control whether or not we are overweight. You mix that with the environment, with diets, stuff like diet and exercise. That's what's going to say whether these genes are turned on or off. If they're turned on, it goes one way. If they're turned off, it goes the other way. Each gene is different. Genome-wide association studies are used to identify common single nucleotide polymorphisms, the SNPs, SNP variations associated with a specific phenotype, association between the inheritance of certain haplotypes, combinations of SNPs that are inherited together, and a specific phenotype suggests that a gene controlling this phenotype may be located near this chromosomal region. So scientists put together a study. They study SNPs. Some SNPs are inherited together, means they're closer on the chromosome. They're less likely to get crossed over during prophase 1 in meiosis. If they're close together, they stick to the other genes that are around it. Certain haplotypes, where the certain SNPs are in the genome compared to 4 different people. Scientists put all this in a computer and study it on a larger scale. And then genetics of human height. Height is complex, involves many genes. It has a heritability of 80 to 90%. Which means you inherit your height, 80% of your height, 80 to 90% of your height from your parents. But over 200 genetic variants contribute to growth and adult height. Account for only 5 to 10% of phenotypic variability. Height is also related to disease. Mutations in some genes result in abnormally short or abnormally tall people. Investigation of new pathways of growth regulation can lead to research on growth disorders. There are really tall people that suffer from hormone disorders, and too much of that hormone is making them really tall, and it causes problems. And skin color is a complex trait. Phenotypic variations in skin color are determined by the amount, type, and distribution of melanin. Melanin is the pigment that makes up our skin color. You have a lot of melanin, you're darker. If you have a little bit of melanin, you're lighter. The more recessive alleles, the lighter the skin color. The more dominant alleles, the darker the skin color, just like eye color. There are some environmental contributions. Variations in skin color are thought to represent the adaptations to the intensity and ultraviolet content of sunlight. And skin color varies with latitude, but not longitude. That makes more sense when we look at the map on the next slide. So the key higher numbers represent darker skin color. You can see there's the equator. And the people located close to the equator have developed more melanin in their skin to protect themselves from the harsh UV rays. And that's all that is. That's all skin color is. It's how much melanin you have in your skin. And then human intelligence is a complex trait. Can intelligence be measured quantitatively? Early studies believe that physical dimensions of regions of the brain were a measure of intelligence. So in the picture, scientists cut up the brain into different regions. And if certain regions were bigger than others, they classified that as intelligence. But they didn't really know how to measure that. So heritability of IQ is between 0.6 and 0.8. So you are 60 to 80% likely to inherit your intelligence from your parents. There is a genetic component. But heritability decreases in monozygotic twins and siblings raised apart, which means there's also an environmental component. So it's a lot of genetics, but there is some environment thrown in there. A lot of your different traits are both a mixture of nature and nurture, and not one versus the other.